Dimethyl, 120 light-years away, a telescope the size of a tennis court locked onto a distant planet, found something that made scientists stop mid-sentence. The molecule it detected is called dimethyl sulfide. On Earth, it is produced almost entirely by living things, by marine phytoplankton blooming across the ocean surface, by microorganisms going about the quiet business of being alive. As far as we know, nothing on Earth makes it without biology. And JWST found a signal of it in the atmosphere of a planet we have never visited, orbiting a star we can barely see. Now, before you reach for a conclusion, here is the first thing you need to know. The scientists who made this discovery did not announce life. The research team, led by astronomers at the University of Cambridge and published in a peer-reviewed journal in 2023, described the signal as tentative. They called for more observations. They were careful, measured, and precise. The headlines were not. Within hours, possible sign of life was circling the globe. Within weeks, other scientists were pushing back, hard. This documentary is about what actually happened in that gap between the data and the headline. What the telescope genuinely found. Why serious researchers disagree about what it means. And what this entire debate reveals about the most important question our species has ever asked. Are we alone? We will tell you clearly what is fact, what is theory, and what is speculation, every step of the way. Before we can judge the signal, we need to understand the planet, and K2-18b is unlike anything in our solar system. In 2015, NASA's Kepler Space Telescope was midway through a second lease on life. Its original planet-hunting mission had been cut short by a mechanical failure, but engineers improvised, and the extended program, called K2, kept the telescope working by using the pressure of sunlight to stabilize it. It was during this improvised mission that Kepler spotted a faint, rhythmic dimming in the light of a small, cool star about 120 light-years from Earth in the direction of the constellation Leo. That dimming was a planet passing in front of its star. The planet was catalogued as K2-18b, and at the time it was just another entry in a growing list of distant worlds. But K2-18b refused to stay ordinary. The planet falls into a category astronomers call a sub-Neptune. It is significantly larger and more massive than Earth, but smaller than Neptune. Here is why that matters. Our own solar system contains nothing in that size range. We have rocky planets and we have gas giants, with a gap in between. Sub-Neptunes sit squarely in that gap, a class of world we have no local example of. Means every assumption we make about them is, to some degree, a guess built on physics and mathematics rather than direct experience. What K2-18b does share with Earth is location. It orbits within its star's habitable zone, the range of distances where liquid water could theoretically exist on or near a planet's surface, given the right atmospheric pressure and conditions. That word, theoretically, is doing a lot of work in that sentence. Being in the habitable zone does not make a planet habitable. It is a necessary starting point, not a conclusion. The star K2-18b orbits is a red dwarf, smaller, cooler, and dimmer than our Sun. Red dwarfs are the most common type of star in the galaxy, and the fact that many of them host planets in their habitable zones makes those planets statistically important targets in the search for life. Now here is where theory enters the picture, and it needs to be clearly labeled as such. A group of researchers, including the Cambridge-led team, proposed that K2-18b might belong to a hypothetical class of world they called a Hycean planet, a world wrapped in a hydrogen-rich atmosphere sitting above a deep global ocean of liquid water. The images you may have seen of K2-18b, a vivid blue ocean world glowing beneath a rust-red sky, are artist illustrations of this hypothesis. They are not photographs. No telescope has ever seen K2-18b's surface directly. Hycean worlds are theoretical. No confirmed example exists anywhere, and that uncertainty sits at the very foundation of everything that came next. The planet itself is strange enough, but the telescope that studied it is even more extraordinary, and what it found in K2-18b's air is where the real controversy begins. To understand what scientists found at K2-18b, you first need to understand the tool they used. The method is as remarkable as the result. The James Webb Space Telescope launched in December 2021, and it carries one capability that changes everything for the search for life. It can read the chemistry of a planet's atmosphere from over 100 light-years away. The technique is called transmission spectroscopy. When a planet passes directly in front of its host star, a tiny fraction of that starlight doesn't get blocked. It skims through the thin shell of the planet's atmosphere instead. Different molecules absorb different wavelengths of that light. Carbon dioxide leaves one signature. Methane leaves another. Each molecule acts like a fingerprint, and JWST's instruments are sensitive enough to lift those prints from starlight. Has traveled across an almost incomprehensible distance. In 2023, a research team led by Niku Madhusudan at the University of Cambridge published results in the Astrophysical Journal. Letters using exactly this technique on K2-18b. What they reported was striking on multiple levels. First, the confirmed findings. The team detected clear, strong signals of carbon dioxide and methane in K2-18b's atmosphere, both carbon-bearing molecules. Crucially, the data also showed unusually low levels of carbon monoxide. That specific combination, carbon dioxide up, methane up, carbon monoxide down, is precisely what some atmospheric models predict you would find above a liquid water ocean on a Hycean world. This part of the finding was considered robust by the team and has held up to initial scrutiny from the broader scientific community. It is, on its own, a genuinely significant result for planetary science. But then came the second finding. And this is where the headlines took over. Embedded in the same spectral data, the team identified what they described as a tentative signal that could be attributed to dimethyl sulfide, or DMS. On Earth, DMS is produced almost entirely by marine phytoplankton and certain other microorganisms as a byproduct of their metabolism. No significant non-biological source of DMS has been identified in Earth's environment. That makes it what astrobiologists call a biosignature candidate, a molecule whose presence could, in principle, point toward biology. Here is where precision of language becomes essential. The researchers themselves were careful. They described the DMS signal as tentative. They stated explicitly that it required confirmation through additional observations. The signal-to-noise ratio for the DMS detection was not at the threshold that astronomy typically demands before treating a result as definitive. The paper did not claim that life had been detected. It described DMS as a candidate biosignature in need of follow-up. Nothing more, nothing less. The media, understandably, found that nuance difficult to contain in a headline. What the Cambridge team had actually produced was a layered result. A solid atmospheric detection of carbon molecules consistent with a theorized ocean world, wrapped around a far more fragile whisper of a molecule that, on our own planet, only living things are known to make. Two findings, two very different levels of confidence, reported in the same paper. So the telescope found carbon molecules, and a ghost of something that might be DMS. But here's where the story fractures. Other scientists looked at the same data and saw something very different. The Cambridge paper had barely cleared peer review before the responses started arriving. Within weeks of publication, independent research groups around the world were poring over the same JWST data, and several of them were not convinced. The first and most pointed objection was about the DMS signal itself. A number of atmospheric scientists argued that the detection was simply too weak to be taken seriously as evidence of anything. In astronomy and physics, confidence in a measurement is expressed in units called sigma. The gold standard for a genuine detection sits at 5 sigma, meaning the odds of the signal being a statistical fluke are less than 1 in 3.5 million. The DMS signal in the 2023 paper fell below that threshold. Some researchers suggested it could be an artifact of the way the raw telescope data was processed. A ghost in the numbers rather than a fingerprint in the atmosphere. This is a legitimate, ongoing dispute, and it has not been resolved. But the skepticism went deeper than statistics. Some atmospheric modelers raised a more fundamental concern: that K2-18b's own chemistry might be capable of producing signals. look biological without involving any biology at all. The planet sits beneath a thick, hydrogen-rich atmosphere, and at those pressures and temperatures, chemical reactions can unfold in ways have no parallel on Earth. In theory, purely abiotic processes, meaning chemistry with no living organisms involved, could generate compounds that superficially resemble biosignatures when filtered through a telescope's instruments. This remains a theory, not a confirmed alternative explanation, but it is a serious one that the field has to grapple with. Then there was a third line of criticism, aimed not at the signal, but at the entire framework used to interpret it. Some scientists pushed back on the Hycean world model itself. K2-18b is significantly more massive than Earth, and at that scale, the pressure at the base of a thick hydrogen atmosphere would be immense. One school of thought holds that any water layer beneath such an atmosphere would not exist as a calm, life-friendly liquid ocean. It might instead be a supercritical fluid, or compressed into exotic high-pressure ice. If the ocean isn't there in the form the model assumes, the biological story built on top of it becomes much harder to sustain. Again, this is a theoretical objection, not a settled verdict. What is important to understand is what this debate is not. It is not scientists versus dreamers. Both sides of this argument are professional researchers working from the same data with rigorous methods. The critics were not saying life is impossible on K2-18b. Many of them explicitly acknowledged that the carbon dioxide and methane detections, the less contested part of the paper, were genuinely interesting and consistent with an unusual atmosphere. Their objection was specifically and carefully aimed at the DMS interpretation. It is equally worth noting that some voices in the broader science communication world overcorrected in the other direction, treating the pushback as a complete debunking of the entire finding. That is also an overreach. The carbon signals remain real. The planet remains a compelling target. The question of what produced those signals remains genuinely open. This is what science looks like from the inside. Not a clean announcement, but a sustained argument conducted in public, with evidence as the referee. The critics raised real questions. The original team wasn't finished, and JWST still has years of observation time ahead. Which brings us to the question that actually matters. What would proof look like, and are we anywhere close to it? Let's start with the numbers, because they reveal just how far the DMS signal was from the finish line. A 1 sigma result could easily be random noise. A 3 sigma result starts to look interesting. But the gold standard, the threshold that separates a real discovery from a statistical coincidence, is 5 sigma, less than a 1 in 3.5 million chance of being a fluke. The DMS signal reported in the 2023 paper did not reach that threshold. The researchers said so themselves. That isn't a scandal, it's a starting point. But it does mean the signal is better described as a whisper than a shout. More JWST observation time is the most direct path forward. As the telescope accumulates additional transits of K2-18b, each time the planet passes in front of its star, the data stack up, the noise averages out, and weak signals either solidify into something real or fade away. That process is already underway. What those follow-up observations will show is genuinely unknown. Anyone who claims otherwise is guessing. But here is something researchers broadly agree on. Even across the debate, no single molecule is going to settle this. The more rigorous view, shared by many in the astrobiology community, is that a convincing biosignature case would need multiple co-occurring molecules, a chemical ensemble that is collectively difficult to explain without invoking biology. DMS alone, even at 5 sigma, would not be enough. You would want to see it alongside other markers, in proportions that abiotic chemistry struggles to reproduce. That is a much higher bar, and it is the right bar to set. History gives us a reason for that caution. In 2020, a research team announced the detection of phosphine in the atmosphere of Venus, a molecule that, like DMS, has known biological sources on Earth. The announcement generated enormous excitement, but subsequent reanalysis by independent groups significantly reduced the confidence in that detection. The debate over Venus's phosphine has not been fully resolved. It remains an open and contested question in planetary science. K2-18b's DMS signal is living in that same uncertain territory right now. And then there is a deeper problem lurking beneath all of this, one that even a perfect 5 sigma DMS detection would not fully solve. If we found an unambiguous biosignature signal, how would we rule out contamination? How would we be certain it wasn't some exotic abiotic chemistry we haven't yet imagined? How would we know it was life as we define it, rather than something chemistry-like that mimics life without actually being alive? These are not hypothetical worries. They are the actual frontier of astrobiology. And they tell you something important. The search for life beyond Earth is not going to end with a single headline. It is going to be a long, careful, contested accumulation of evidence, exactly like every other major discovery in the history of science. Step back for a moment from the molecule. Step back from the telescope, the spectral graphs, the sigma thresholds, and the competing papers. Because the K2-18b story, at its core, is not really about whether one particular planet has life. It is about something larger and stranger than that. It is about the moment humanity first built an instrument sensitive enough to make the question real. Before JWST, the atmospheres of small, potentially habitable worlds were essentially invisible to us. We could detect giant planets, hot gas giants close to their stars, worlds nothing like our own. But a sub-Neptune sitting in a habitable zone, 120 light-years away. That atmosphere was beyond our reach. Now it is not. The fact that scientists are arguing, genuinely, rigorously arguing, over the interpretation of a real chemical signal from a real planet at that distance is, by itself, a historic milestone. The argument is the achievement, and that argument is healthy. The pushback against the DMS interpretation is not a failure. It is not a scandal. It is science doing exactly what science is supposed to do, applying maximum pressure to extraordinary claims before accepting them. Every objection raised by independent researchers, every alternative model proposed, every demand for higher statistical confidence, that is the immune system of knowledge working correctly. A claim that survives that kind of scrutiny means something. A claim that doesn't survive it gets refined or discarded, and either outcome moves us forward. Zoom out further still. K2-18b orbits one star among an estimated 200 to 400 billion in our galaxy alone. A rough figure, but one that staggers the imagination. Most of those stars are red dwarfs. Many of those red dwarfs almost certainly host planets in their habitable zones. If life can emerge under Hycean conditions, in a hydrogen-rich atmosphere above a liquid ocean, then the universe may be saturated with it in ways we have barely begun to contemplate. And if it cannot, if Hycean worlds turn out to be sterile despite their oceans, that answer is equally profound. Either way, we learn something fundamental about where we stand in the cosmos. To be completely honest, as of now, no confirmed detection of extraterrestrial life exists anywhere. Not on K2-18b, not on Mars, not anywhere we have looked. What exists is a tentative signal, a live scientific debate, and a telescope that will keep watching. That is not nothing. That is, in fact, everything we could reasonably hope for at this stage. The reason this story travels so far, so fast, is not because scientists found aliens. It is because for the first time in human history, we have a machine in space capable of turning the question of life beyond Earth from philosophy into measurement. The question is no longer purely abstract. It has a spectrum. It has data. It has disagreement, which means it has a future. The next JWST observation window could shift this debate entirely. When it does, we will be here to cover it. Until then, the question stands open, and that is exactly where good science lives. The universe has not answered yet. But for the first time, we are close enough to hear the silence, and wonder, genuinely wonder, if something somewhere is about to answer back.